Hello friends, today I am going to talk about the various types of cell-based assays and their principles. First, I will talk about the sulfur-rhodamine B assay. It is used for the cell density determination. Sulfur-rhodamine B is a anionic dye. It will bind to the cellular protein in the acidic condition, but in basic condition it will dissociate from the proteins. So in this assay we generally add the sulfur rhodamine B dye with the cells, and we will add 10% this trichloroacidic acid for the fixation of the cells. After that we will see the intensity of the sulfur rhodamine in alkaline condition, and its intensity will be directly proportional to the viability of the cells. This assay we will see the absorption at 520 nanometer, and it is used for the drug induced cytotoxicity studies, and it is also a non-destructive kind of assay. Next is natural red dye assay. In natural red dye assay, it's what will happen when we will add to natural red dye to the cells. It will go inside the cells and accumulate in the lysosomes in the acidic conditions, and after adding the acidified alcohol, dye will be solubilized in this alcohol and cell will rupture, and then dye will come out, and this intensity will be directly proportional to the viability of the cells. Next, MTT assay, which is most sensitive assay. It is used for the metabolic activity and determination of the cells. In this assay, what will happen when we will add this MTT? MTT is nothing but tetrazolium dye. It will go inside the cell, and it will convert into the formagen in the presence of NADPH. This NADPH simply reduce this MTT into formagen, which is purple in color. When this formagen is formed, it will be crystal shaped, insoluble in nature. So when we will add a DMSO, which will solubilize this formagen, and it will give the purple color solution. Absorption will be at 500 to 600 nanometer we get, and if the higher the concentration of the formagen, higher will be the intensity of the color, and we will get to know that higher will be the cell viability. Next is XTT assay. It is alternate assay of the MTT assay. It is said that it is more sensitive than MTT assay. What will happen when phenyl methyl alcohol and XTT is incubated with cells? This phenyl, in the presence of phenyl methyl alcohol, mitochondrial dehydrogenase will convert this oxidized NADPH into NADPH, and next this oxidized in this reduced NADPH will convert extracellular XTT into formagen, this which will be orange in color, and this is soluble crystal. Here in MTT, it will formagen is producing inside the cell and insoluble in nature, but here in XTT, formagen is producing outside the cell and soluble in nature. Hence in this XTT assay, this solubilization step is not required. In this XTT assay, we will get to OD at 450 nanometer and 660 nanometer, and this color formagen produced color will be directly proportional to the number of viable cells. Next is Resajurin assay. It is also known as LMR blue assay. It is used for the cell viability of the cells and to determine antimicrobial activity of the cells. In this assay, what will happen? This resazurin oxidized in nature will be reduced to resorufin, which is reduced form, and it is pink in color, and it will give fluorescence signal which will be proportional to the live cells. Here this fluorescence excitation wavelength is 530 to 730 nanometer and emission wavelength is 580 to 590 nanometer. Next is protease viability marker assay. Protease activity disappears after the cell death. So protease is a good marker of live cells. In this assay, this aminofluorocoumarin compound is incubated with the cells. In cells, this aminopeptidase enzyme will break down this amino moiety and AFC moiety separately, and this fluorocoumarin will give fluorescence which will be proportional to the live cells. Here this lambda emission will be 380 to 400 and lambda emission will be 505 nanometer, and using fluorometer. Next cell-based assay is calcine AM assay. In this, calcine and AM will be produced by the enzyme esterase, and it will give fluorescence. Luminescence-based assay using ATP is produced luciferase and the presence of ATP.